All right, so neurons and glia. This is our last set of slides. In order for us to understand the types of functions that happen, we need to understand the types of cells in charge of those functions. So if you can remember, neurons are the ones that are in charge of the electrical components, whilst the glia are more the supportive type of cells. So though they will not be the key focus of our physiology classes, they're just as important. Well, not just as, but they are important. So let's take a review. So the neurons themselves range from a few millions in variation, with in terms of length, and the longest being from the toe to the base of the neck. I know, right? So, good fun fact. So, anatomically, they differ in appearance, and in physiologically, they also differ in terms of are they inputs or are they outputs. A type of excitability, how excitable they are, is also different in terms of firing rate. Do they fire quickly? Are they very sensitive? They also differ in that vein. And these things are things that we introduced to you in physiology 1 in your second year. Uh, they are mostly for communication, and they communicate using two methods. They can communicate just by this anatomical type of linkage that they have, where the depolarization of one neuron is going to sort of anatomically pass on to the next one, and this is called the electrical transmission, while the chemical transmission is more or less the polarization causing um, a release of a chemical at the terminal end, and then this synaptic activity causes the next neuron to also undergo depolarization and then have that action potential reinitiated in the next neuron. So those are the things that I would like to review. But anatomically, we want to remember that we have the nucleus surrounded by, within the soma or the cell body, and it has these dendritic ends, which are more of the receiving components of the neurons. And then we do have these axons, which can be myelinated or non-myelinated. And if they're myelinated, we see that the action potential moves only in these spaces that are not covered by the Schwann cells. And this type of, saltatory movement on the nodes of Ranvier only makes this movement much faster than if it had to go through, each space of the axon. These are things that we learned about. But if you do not remember, we could still spend some time in our next physical class where I can re-explain some of these things until the activities reach the terminal end, and we have the next part being excited or inhibited, depending on what type of neuron it is. The glia cells, on the other end, called support cells, and we have oligodendrocytes and astrocytes. The astrocytes are really for cleaning up. They more or less pick up excess neurotransmitters, and that is more like downplaying what they do. But this is just speaking broadly. While the myelin sheath is made up of oligodendrocytes and Schwann cells, depending on which part of the central nervous system they're from. So we see that microglia are 5% and are immune. cells. So they react to sort of rectify damage that an infection to the neuronal components. So demyelination may cause multiple sclerosis in the CNS, and this is demyelination oligodendrocytes. So we're not talking about CNS, we're talking about oligodendrocytes. While GB is demyelination because of the Schwann cells, and the Schwann cells in the peripheral nervous system. So please make sure not the oligodendrocytes are inside, and the Schwann no, they just on the peripheral end. Um, so even the types of diseases due to their demyelination that they cause different names. So multiple sclerosis, oligodendrocytes, then Guillain-Barré, we have Schwann cells. Okay, so let's try to think, pair, and share. Number one, review the speeds in different types of neurons from Ganong. Then review the mechanism of action potentials in neurons, again from Ganong. And then review the types of synaptic ends or junctions. And then when we come back, we're going to go through these components. So this think, pair, and share is long term until we meet again. Okay, just a few more facts before I end today's class. I'd like you to be very uh, observant on the notion of summation, which can be temporal or spatial. Now, when we talk of temporal summation, which is a result of repetition of afferent stimuli, you know, um, they cause this new set of excitatory postsynaptic uh, potentials before the previous one could have decayed. So you see that just before the first one moves out, another one comes in, and you have this temporal summation that happens because they're coming in at a fast rate. Okay, when the activity present is coming from more than one knob, then you have multiple sources of action potentials to one cell. That is known as spatial space summation. Okay, read about that. When you're talking about the inhibitory component, I want you to look at postsynaptic inhibition, and then how does it differ from indirect inhibition? What is the difference between tonically active neurons, and how do they differ from phasic ones? These questions that I am asking you to review or point out from your first year are really a good foundation to understanding some of the things that we explain in neurophysiology. So make sure that you go through this work. And when we meet again, we will rewind and just have a lesson that will start with action potentials and end at the synaptic junction before we start looking at receptors. Okay, so this is just a slide to really just go through everything that I've said um, concerning the neuroglia. So it talks about which ones are in the central nervous system and which ones are in the peripheral nervous system. I'll give this to your class rep so that you could have it handy. In your next class with, I think, our lab that's in the power lab, you're going to look at the reflexes. So also make sure that you read up on that before you go for this class, and then we'll meet. Maybe something that will be a take home is, if someone has a larger reflex than normal, what do you think? Is it a sensory or a motor impairment? See you next time.